This is Joanne Geer from the Biopharma Research Council. We'll wait just a couple of ticks to get started, and but just a little moment to let everyone get logged in. All right, well, good morning, everyone, or evening, wherever you are, or very early morning, if that's the case. This is Joanne Geer. I'm Executive Director of the Biopharma Research Council. We are an association for scientists across all the different silos of biomedical research, which includes industry, academia, nonprofit, government, and supplier labs and their teams. We work throughout the year to provide educational programs, many of them as webinars like this one, and also several conferences throughout the year. I really like this slide. This is from a conference we did last year, and that's Bob Boland from Janssen Pharmaceutica. And our whole mission is to help scientists talk to each other across all those different silos. I used to say develop a common language, but we don't need a third language. We need to learn each other's languages. There's some of the activities that are happening. We just, about a week ago, we had a really wonderful CRISPR conference, Advancing Drug Discovery Through Genome Engineering in Boston. We have an occasional series, webinar series on global collaboration to address Ebola, and Pardis Sabeti did a wonderful webinar for us a little while ago. We just finished a short course in the microbiome, and in September we're going to the NIH and having a full-day program there, which is called Manipulating the Microbiome, Can It Impact Health? And we have coming up at the end of July in Princeton is the Internet of Medical Things, Cybersecurity and Discovery Development and Devices. And it's focused, there's a two-day piece. The first day is more of a workshop for IT professionals, and the second day is really for everyone who manages precious data. I'm just sharing a little bit about this theme of data to drugs and diagnostics, of which today's webinar is a part. As we all know, we can work with data from any part of the system and anywhere in the world. This was data we collected on our audience at a conference we did last year, and we'll be revisiting this topic from time to time. We asked people on a continuum of all science to all IT, where do you put yourself? And I think it's pretty interesting. We had 44% of the people who were equally saw themselves as scientists and as IT professionals, and I think that's quite significant and a growing cohort. It's not the only topic that we address on an ongoing basis, but it's a very important one, and we really appreciate our speakers today in helping us chew on the subject. Previous webinars in this series are available on our website, so I won't belabor those. And here we are today. So our session today is on dealing with disparate data. I'm going to introduce all of our speakers in the order of appearance, which is not quite the same order as you see in front of you, and I'll let you know who's who. We will first be hearing from William Logging, who is the head of production bioinformatics and associate professor of genetics at Mount Sinai School of Medicine. Dr. Logging has spent the last 20 years focusing on generating treatments for human disease, bridging both clinical genomics and pharmaceutical sciences. He has been globally recognized for his work with Cellzentry for use in HIV. The results of his contribution to the approval of Jardiance were listed within the drug insert. He has been published in several leading scientific journals on the utilization of novel informatic approaches to drug discovery, and his TEDx talk last year on cancer genomics was highly acclaimed. He currently holds several appointments at Mount Sinai School of Medicine, where he focuses on the clinical fields of oncology and immunology. So that's William Logging, who is the second from the right in your picture. Next, we'll hear from Tim Miller, who's on the left. He's the Vice President for Platform Strategy at Thomson Reuters. In his current role, Tim is responsible for the life sciences platform, focusing on developing new ways for customers to derive additional value from their content, including enhancements to the CORTELLIS web portal, web services APIs, informatics, and visualization technologies. Tim holds a bachelor's degree in chemistry from the University of York and a bachelor's degree in law from the University of London. He is a chartered chemist, member of the Royal Society of Chemistry, and a chartered information technology professional, member of the British Computer Society. Thirdly, we will hear from Andrew Garrow, who is a director at Thomson Reuters. Andrew is a data scientist in Thomson Reuters IP and Science Division. He is focused on the CORTELLIS Data Fusion program, a data integration solution powered by a unique hybrid of big data and semantic technologies. Before working on CDF, Andrew developed capabilities to text mine clinical data with CORTELLIS and methodologies for mining social media for pharma relevant information. Now, Dr. Earhart will be joining us after we hear those three speakers, and I've invited her as an interested party who's new to the discussion, and she will be participating in the Q&A at the end and helping us all to query further into the topic. She is the director, so Dr. Anke Earhart is Director of Clinical Flow Cytometry at Bristol-Myers Squibb. As head of a technology core facility in a Fortune 500 pharmaceutical company, she has developed and managed a large number of successful collaborations. As a member of our Biopharma Research Council Scientific Advisory Board, she brings her experience as collaborator and leader and as a subject matter expert in imaging and flow cytometry, together with her enthusiasm for driving connectivity in biopharmaceutical research. I want to thank everyone for being here today, and I want to invite you all to enter questions at any time in the course of the sessions. If there's a specific slide you're interested in, if you'll note that in your question, that will be great. We'll address all of the questions at the end of our three talks. And I will now introduce Dr. William Logging.
Okay, can everyone see my screen okay?
I don't think we're quite there yet.
Okay. Thanks for that very nice intro, Joanne.
Well, thank you. You know, it's really terrific to have a diverse environment like this and diverse speakers to be able to really look at different sides of the question.
Okay, yes, we do see your screen.
Okay, thank you very much. So today what I'm going to really try to briefly focus on is where are we at as far as the technology of being able to interpret a patient's genome, whether at the DNA level or at the RNA level. And I wanted to just start off with a really interesting perspective, because whether we know it or not, we're actually entering what I think is sort of the golden age of healthcare and being able to interpret one's genome. But this is sort of a step-wise. And so about 15 years ago, sequencing technologies, obviously we were sort of using very early types of approaches. Some of you may be familiar with SAGE. I actually performed the SAGE technique in the lab many, many times. And about 15 years ago, you had to actually sequence around 30,000 base pair to get an adequate read of a SAGE experiment. And we found that that would usually take us, even if we were to send our libraries to WashU to be sequenced, it would take roughly around 3 months for that data to come back. Today, we can go right down the hall, put our samples in a next generation sequencing machine, and roughly get about 20 to 30 million reads per sample. And I list here less than a week, but obviously you could do that literally within 48 hours. You could even get more than 20 to 30 million reads depending on how you load the flow cell. So again, this technology has really rapidly progressed in just the space of 15 years. And what's very exciting about it is, you know, we feel, and obviously we hope others would be along with the same thought process, that the DNA sequencing technology will change the course of human history. And obviously what we're seeing here is the rapid progression of the lowering of the cost to be able to sequence 1 million nucleotides. I originally in this slide deck had the overlay of Moore's Law, but I did not want to show that because we've probably all seen that and we could have all just drawn that from memory. But one of the really exciting things is, again, the cost of sequencing one genome has dropped tremendously, very highly, even within the course of the past 10 years. Now, if you look at this slide and we say, yeah, it's dropped, that's very amazing, it's all pretty exciting, but what if we were to apply the same type of rapid progression of technology advancement to other industries? What would that look like? And so I took the liberty of actually looking at other industries. And so I'm just going to briefly focus on two, the automobile industry, specifically Tesla. This is not to pick on Tesla, I actually like the company. And then also Boeing with their 787. I've been told that this is what first class looks like in the Boeing 787. So if we were to apply that same progression of technology from sequencing to the Tesla, a $100,000 Tesla automobile today would, in 15 years, in 2030, be able to travel 500 times further than the current model. Okay, so we look 500 times. Yeah, that's great. That actually comes out to about 240,000 miles per charge. That's pretty impressive. And this whole entire automobile would cost you roughly 33 cents. That is equally very impressive. What if we were to place this on to the Boeing 787? If we were to apply that same technology, they would be coming out with a airliner that in 2030 would be able to circumnavigate the globe about 150 times on a single fuel tank, and quite interestingly be able to fly direct from Shanghai to New York in less than 2 minutes. But when I discussed that with my colleagues, they felt, well, it won't take just 2 minutes. You've got to get through security and so forth. So obviously it's going to be a little bit longer than 2 minutes. But all joking aside, this rapid progression of technology and these tools, we want to apply them to the biology field. And what we're seeing, and very seriously now, biology is very complex, and we have to approach it with a lot of humility because there's a lot we know, but there is a lot we definitely do not know. And so historically, a lot of this data has really been applied, and these types of early approaches have been applied to the drug discovery process very early on. And what we're seeing, targets and disease information, the return on investment was often quite low, and that made a lot of people question, okay, do these technologies really even are supportive to do the things we want to do within an organization? What we're seeing is now that we can apply these technologies later on, where patient stratification, disease repositioning, drug asset differentiation with competitors to standard of care, all very important within the drug discovery process. And now if we can apply these technologies in this space, we're getting a lot bigger bang for a buck and as far as return on investment. Just briefly, I'm going to step through this. I don't want to go too in-depth into the science, but just to give a quick example, how does these techniques and technologies impact specifically drug discovery? If you've got a clinical asset and you're trying to take it to the FDA, you really want to show differentiation with standard of care. You want to show that you're better than the standard of care, obviously for the pay system, but you also want to show that you can differentiate from competitors. You can apply these technologies to subjects that are dosed with different tissues or with different actual preclinical drugs and drugs that one would want to take through the clinic to dosing subjects. You can perform RNA-seq and actually take that data and map it to pathway differentiation. And so just a quick example, this is actually using some software that we have internally, the ASD Cure software, where we looked at several thousand pathways, the genes that changed across a standard of care on the x-axis as we increased in dose. And we basically saw the pathway activity on the y-axis did not change when we increased the dose of the standard of care. However, when we did take the candidate and applied that same analysis, we actually had a significant p-value and a drop of the pathway activity, which was saying in this plot that the candidate, the drug candidate, is actually turning that pathway off as you increase the dose. That's pretty significant because now you can take in your data package to the FDA and say, wow, I can differentiate between standard of care or competitors. My competitors are doing this, but my drug's actually doing this, and we think that it makes it a better choice to go into patients and actually ultimately get approved. And so these are the types of things that we would like to be able to and continue to do with this type of technology. These technologies have been used in a wide array of other areas. I'll just briefly touch on those. past experience, personal experience that I've had using these technologies. We've used them in submission for Maraviroc, for the NDA. We've actually taken these technologies and used them for the approval process for empagliflozin, and the results, as well as other studies that were in those approaches, were listed in the drug insert. And then another example where we had these technologies and we've actually obtained results that we were able to patent. So a wide array of different types of technologies within the omics field applied to real learnings within the drug discovery space. Again, how this technology impacts the patient is that we've been able to recently receive an IND approval from the FDA to go into vaccine therapy for oncology, and that's a pretty exciting path that we're going to be taking at the Mount Sinai system. One of the things I just wanted to really highlight, a number of the investigators that were listed in that IND, as well as others that went to the FDA, and again, we just recently received approval on that. I just want to highlight one individual, my colleague Jeff Hammerbacher. If those of you who do not know him, Jeff is a pretty interesting individual. He originally was one of the founding data scientists at Facebook. And so my sort of comment is, how many times do you see an FDA-approved IND that has someone with those types of credentials? Obviously, the types of data and the data ecosystem that we're using internally within our system at Mount Sinai, very large, several petabyte worth of storage. This is just the data side to store the actual data. Now we've got the compute core is well over 12,000 core and approaching some 10 petabytes worth of raw storage space. In the past, you would have thought about this type of technology and you would have said, wow, this is something like should be in other industries, should be in obviously something having to do with telecommunications or any other type of high compute industry. But we're applying that now to the genomics field, and those mergers are going to continue to take place. What drives us, what drives discoveries, obviously, is using these novel tools and seeing something new. I liken it to Galileo. In the past, 1610, what were people doing with telescopes? They were using it basically to spy on their neighbors. Galileo took his, pointed it at Jupiter, saw these dots flying around Jupiter, made some really interesting notes, and basically what he was able to discover was the Galilean moons, which are obviously in the news as we're planning to send robotic missions to those moons. We see this as a really large opportunity to be able to take the wealth of this information, apply it at the genomics level, data visualization, analyzing all these billions of data points. But of course, we need individuals thinking in a scientific standpoint. These concepts obviously are explored further in an upcoming book that I have coming out towards the end of this year, and I've been fortunate enough to be able to work with a number of other contributors who put chapters in, and I'm very pleased to say that we explore a number of different topics, take the reader through the entire drug discovery process, and then state with a lot of examples as well as use cases how one could apply these types of techniques to the drug discovery process. Again, thank you very much for your attention. It is very exciting to be in a group of individuals that have these types of backgrounds, and I look forward to, again, stimulating discussions. Thank you very much.
Okay, thank you very much, Will. I'm just going to, it tells me I'm sharing my screen, so I shall put it up. Is that good? Okay, yep. So I'm going to, thank you. I'm going to sort of take another direction at the same sort of problem. As Will explained very clearly, the amount of exciting and the millions of terabytes of data coming out of NGS, that's not the only place where we can see more and more data coming out that we have to deal with. So I'm going to sort of look at one of the other sources of disparate data, one that is probably even earlier in its life cycle than than the one Will was talking about, which is the role of devices held by patients. So the whole wearable technologies and the uses thereof in clinical trials and in consumer health. And what we see there is a really interesting cycle. You see in the center there, companies are beginning to realize that the enormous power you can get from collecting information from patients, and you can find information about those patients, whether they're in a trial or whether you're looking at compliance in their normal day-to-day life, taking tablets. But they're interested in things. Either the interest is coming from the scientist who's trying to find out information, or that's coming from the patient trying to understand his own particular disease and his own particular version of that disease, because as we know, no two diseases are really the same. And what we're beginning to see is the evolution of tools to take advantage of these wearable technologies. We see smart application developers building devices for these patients, and then we see the consequence of that, which is a wave of data that we have to deal with, the data that comes out of these devices and the analytics that you can apply to these devices, to the data that comes from these devices, so that you can actually try to get insight from it. And that's really interesting from our perspective as a provider of information, because we're seeing a change in what people need from us. So I've put the big 3 arrows on this. First of all, there's much more targeted information that needs to get to the patient, to the person who is wearing the device. You can't put sort of Encyclopedia Britannica on the Apple Watch. You want to just basically give the person what he actually wants. And then as you come out the other end, this tidal wave of little tiny bits of information needs to be cleaned, needs to be, to go through the rigors of data science, to be cleaned, analyzed, and processed so that you can gain the relevant insight. And so I'm going to talk about those sort of 3 arrows and some of the things that we're exploring in Thomson Reuters and that we've seen from people that are addressing this problem. So I'm going to kick off with looking at the first thing, feeding information to a patient. As I said earlier, the key is to provide the right information and in a consumer-friendly kind of way, and that's a sort of learning cycle for us. We are traditionally dealing with organizations who are health professionals who don't need to be told, don't need to be sort of have stuff explained to them. So we're now having to look at how do we get targeted information in digestible form to those kind of users. So an example here, to explain to a patient what actually is going on in hepatitis D and using multimedia to explain it. Second example is looking at the whole business of personalization of content and filtering of content. Again, as people are using these sort of information and consume this kind of information in a smart way, they need just the right information. They need to be able to get only the information that's actually truly relevant to them. And with a wearable device, with that kind of personalization, you know a lot about the customer because the person wearing the device has already input who they are. With a bit of luck, they would have gone to Will's lab and got their sequence done so that we know exactly what particular variants they have, and you can start to target information to their particular needs. You're no longer giving them the fire hose, you're giving them targeted relevant information. And the third goal, I think, of providing information to an end user is to make it actionable. I liken this particular application we've developed to something that to things like Uber. What does Uber do? It basically tells you that there's a cab around the corner that's willing to take you to the bank or whatever. What we're hearing a lot from customers is that they want to hear is there a clinic nearby that's running a clinical trial for a drug that actually talks to my particular disease, to my particular genetic variation. And so you can see here, just by simply looking at what trials are in progress and the inclusion-exclusion criteria for those particular trials, you can tailor in to find a particular site that's doing something that you may want to be interested in. You may want to actually sort of contact. I see it's Mount Sinai, and they got 3.8 stars. So that's a good one for Will. So yes, so when we're providing information to customers, it's all about getting that data down to really, really tiny little relevant byte chunks. Now I'm going to turn to looking to the other two sort of big orange arrows on my screen, which are the problems of data cleaning and the problems of generating insight. And I'm going to start with the issue of data cleaning. And in the world of data cleaning, and certainly in the world of life sciences, ontologies rock. You need ontologies to make sense of what's coming out, especially in a wearable, consumable world. If you go beyond wearables into social media and places like that, people will not use the right name for things. They will use million names for things. And if you want to try and find connections and linkages between things, you need to have those ontologies. So I made up this sort of fake example here where in the top left-hand corner, you've got a bunch of synonyms for good old aspirin, and you've got a bunch of synonyms for Pfizer and a bunch of synonyms for type 2 diabetes. If you just tracked the particular phrases, maybe somebody says, I'm taking Novid from Upjohn for my DO3924, it's a very, very weak signal, but by the time you've collected a bunch of signals for what is really going on, which is Pfizer, aspirin, and type 2 diabetes, and I would stress this is a completely fake example, you can start to surface up information that would otherwise be completely hidden. You wouldn't be able to make sense of it. You end up with it's like sitting outside the Tower of Babel trying to figure out what's going on. So ontologies really help to make sense of the content, to make it possible to find the different connections between things. And it's not just synonymy. I've focused on synonymy here, but it's also the hierarchy. I might be interested in all the mentions of COX-2 inhibitors in type 2 diabetes, for example, or I might be interested in all big pharma companies who are working with aspirin. Being able to sort of look at that and to be able to helicopter up using the power of ontologies is also part of the package, part of the way in which ontologies really help to answer these kind of questions. And my last sort of topic, and I think, if anything, the two things I'd like people to think about coming out of this is it's linking data through ontologies. It's also about using the data that we already have as context, and to provide context with the data that we already have. I've talked in the past about big data and little data and how the power of little data is to make sense of big data. And I still very much believe that. You imagine you've got all of these signals coming out from all these devices, from all these NGS stuff, all of the social media. A lot of it is going to be junk. A lot of it is going to be too weak a signal to actually be actionable. A lot of it is going to be signals which are perfectly reasonable. Like aspirin's good for headaches, but you don't want to be. You don't need to react to that. That's a known thing. So, first thing that you can do with a universe of good known facts is you can act as a sort of low-pass filter. You can deal with the signal-to-noise problem by looking for things that are different, things that stand out, and you use that information as a filter. And that's what I've done here. I've got a bunch of information, and there's one particular thing that rises its head above the parapet. And in this particular case, it's a variant drug relationship, and that's great. You have taken the unit of information, and you're now targeting on one particular thing, but humans need to sort of go beyond the fact. They want to follow the train of thought from that one fact to so what. It's all about the so what. So once you've got that particular piece of information, you can start asking those subsequent questions you see in the other clouds. And you want to do that in a seamless kind of way. You want to follow your train of thought. You don't want to be sitting there, going to one place to find out whether or not there was a trial, going somewhere else to do a literature search, maybe going to a machine and checking out the variant. You want to be able to sort of follow that through. And I think that's another area where having, first of all, the right data, but secondly, having that data accessible in an easy programmatic kind of way, so you can pull it into whatever analytics package or environment that you want to work with can really help to help people, in particular scientists, sort of go from signal to hypothesis to action. And in my last slide, I'm just underlining that you've now go back onto the top of the circle. You've now got a new hypothesis that something is interesting. You can then go and feed that back around the circle, find out more about it, and move on to further and greater discoveries. And on that, I shall pass you over to my colleague, Andrew.
Hi everyone. So you should be able to see my slides very shortly. Okay. I think you should be able to see them now. Okay, so I'm going to give a presentation on a solution from Thomson Reuters that enables integrated data analytics, and this is something we call CORTELLIS data fusion. We've heard a lot in the last couple of presentations about how the volumes of data that customers are having to we've heard about how the volumes of data people are having to handle is ever increasing, and this brings with it various problems in terms of being able to navigate the landscape of data and connect the dots between all these different data sources that are out there. So if I was to give you a one-slide summary of what our solution looks like, we have here an example from a customer. This particular customer, they had to manage a portfolio of projects. And one of the things they were having to do in this is they were having to measure which of their projects were being successful and ultimately where should they be putting their money next to support future projects. Well, they had lots of ways of measuring success, and they had lots and lots of data to support this. But their problem was that all this data was basically dotted all over the place. Some of their content was private content, so this is information they were measuring internally in their own systems and places. But they also had proprietary content that they were subscribing to and public content that they were able to access. Even more complex was the fact that their content was in a variety of different formats. So they had some very structured format information coming from databases. This could be databases with information on records, or it could be experimental type data sets. They had some unstructured content, so things like reports, very text-heavy type documents. But like many customers, they also had a lot of semi-structured data, so this is like small Excel files and things with vital information, but it's not really the sort of information you've traditionally put within a more formal database. So what these guys were needing was basically a solution where they could connect all this data together so that it's all in one place. They needed the system to be very scalable and performant, off which they could then perform their various search and analytics to address what their ultimate business objectives are. So this is the sort of solution that we build using CORTELLIS data fusion. So what I'll go through in the coming slides is really a bit on the philosophy of the approach about how we build solutions, and I'll talk a very small amount about the technology. So in terms of a use case, my background is actually bioinformatics, and I was in bioinformatics quite a few years ago, so I will focus on discovery science. But the approach can be basically taken to any of these new sorts of sciences, such as patient stratification or personalized medicine, etc. So basically, you have to think that whatever your science area, you're going to have a number of core business objectives that you're trying to solve, or core objectives you're trying to solve. And within this, you can then break it down into subsets of questions. So if you're in discovery, you might want to be able to identify what trials are going on for your disease, what targets are these drugs impacting, what do we know about these targets. And in any one of these questions, you have things, you have entities, you have drugs or trials or diseases that you're interested in. And for any of these things, you have the relationships between these things. And this really is a really important conceptual way of thinking about how you're starting to want to connect your data and bring it all together. As mentioned previously, the data can be coming from lots of different places. So if I was to go into that sort of approach for solving this integrated data challenge and navigating the data, one of your example questions might be, well, tell me about phase 2 drugs for diabetes. Well, the first thing you might want to do is say, well, what trials do we know that are going on for diabetes? And what drugs are associated with these trials? And that's a really simple thing to link together. But actually, often our customers, they don't want to have to go to multiple places to then find out about that individual drug. They want to have all that drug information in one particular place. So what they actually want to then do is to connect all their drug information into one particular record for that drug. The reason why they want to do this is because they then may have information that is then connecting that drug to other resources that they have access to that are separate to where you've linked the trial and the drug. So what we can do now is you can then say connect from your drug to information that you have on projects that are related to that drug. These project files might be in Excel or SharePoint, etc. They might want to be able to link back to assay data sets that they have relating to that drug. So this is ultimately the sort of thing that we're wanting to build in an integrated data solution. Well, that same approach you can take for many different questions. So, for example, what drugs are people working on for my disease of interest? Well, this question can be broken down. You want to connect from a disease to the associated clinical trials, to what therapeutics are those clinical trials based on, and then what proteins are those therapeutics targeting. You can do the same thing for what adverse events may my drug cause, where you're connecting from a compound to a target or pathway, and from there to adverse events, and so forth for lots and lots of different questions. Remember, for any one of these entities that we have within one of these questions, the information for these entities might be coming from lots of different places. Well, this is for a long time. This has been like a real dream solution to be able to solve. But it's always been very, very difficult. And there's a number of reasons why this has always been very difficult. So the first reason why this sort of analytics has always been challenging is basically your data is often scattered across many different places. These places, like I say, they can be internal, they can be on your own intranet, they can be external somewhere else on the web. In addition to that, people are often faced with lots and lots of different data formats and very, very diverse data formats. So you have your Oracle type data tables, but you've also got things like Excel, you've got documents. So how you connect all that sort of very different data has always been challenging. But you have other problems similar to what Tim was actually just talking about in terms of ontologies and how you glue together information. Certainly if you have databases, people might be very strict on the way that they're using IDs and try and use similar IDs. But as soon as you start to integrate different sorts of data, your Excels, your SharePoints, your papers and things, all of a sudden the ideas break down. You need better ways of actually mapping the content together in a much more semantic way. So what we've been able to build with our solution, CORTELLIS Data Fusion, is that we've been able to suddenly start to exploit some of the new big data and semantic technologies that people may be hearing about. So big data is obviously a bit of a buzzword, and we see what sort of an impact big data is having in the next-gen sequencing type worlds. But big data is all over the place. We actually see the volume component of big data as one of the easiest components to solve, because what you can do is you can ultimately solve it with various big data technologies, put more nodes in your computer cluster. Where we've always found that the biggest big data challenges are in terms of the integration and the variety. And for this component, we're within CORTELLIS Data Fusion, we're using a number of semantic data technologies that make this much easier than it's traditionally been. So in terms of that, we also need to be able to make sure that the system is optimized so that you can search different types of content. So you need to be able to search across both your structured contents and your more unstructured text-heavy type contents. The ability to actually integrate documents into a data integration solution is really important, because that helps you avoid something we call the data graveyard, where basically someone has a really important PowerPoint on their laptop and then they leave the company and that PowerPoint or that Excel is gone. So the ability to integrate all this content into one place is really important. Finally, one of the importance of being able to have a data fusion and data integration solution is really around performance analytics. If you're able to bring your data together quickly and into an index like this, then enables much more performant analytics than if your data is scattered all over the place. So second, penultimate slide, but it goes back to one of the things that Tim was saying, that we very much believe that big data aspires to be little data, and this really is, even though you're generating lots and lots of information, you're able to come up with these great statistics, ultimately the clinician or the bench scientist or the discovery scientist, etc., they don't want to see loads and loads of information. They want to be able to get down to the key snippets of information that are going to enable them to address their real needs. And we speak to lots of people that have this particular need, but often they want to see their information in a slightly different format. So a second sort of key philosophical thing that we have is that no one solution fits all. So an API layer is really, really important in any solution like this, because from your API layer, you can then go off and you can build your infographics, analytics, plug your content into workflows. We have with CDF ways of building all sorts of different interfaces. Out the box is a very cool data explorer interface that's it does some basic data search, but beyond that you can start to perform some very cool analytics on it as well. Another thing that I don't think is ever going to go away is actually just the ability to export chunks of content and then get that into something like Excel, where people can start doing other things with that. So big data aspires to be little data, and no one solution fits all. So another thing, if you've got a big data analytics and integration tool, another thing that's really, really important is having some sort of management layer. So I'm not going to go into the data management layer that we have here, but we can definitely do demos on that in other sessions. But really, the important things that you need in this sort of a data management layer are the ability to easily connect new content and then help define how that content should all be integrated. If you're having ontologies in your system, the ability to plug those ontologies into the system and show how they should be working. So just to wrap up, we typically hear from our customers that their biggest big data needs are around integration. Content is key, so ultimately if you're trying to address real business or scientific objectives, it's the data that's going to enable you to get there, so content is key. And then just very finally, semantic technologies and big data are really, really helping us in develop new solutions for this. So thank you very much.
Well, thank you, Andrew, and thank you to all of our speakers. This is Joanne Geer again, and I want to encourage everyone to continue to add questions in through the box on the right. We have some really interesting questions here. But first, Dr. Earhart, would you like to join us? And I don't want to put you on the spot, but I'm going to. Do you have any immediate questions or comments that you would like to share?
Well, I have a bunch of questions, and actually I have one question that probably goes to both Tim and Andrew, because it's taking together what they're talking about and maybe mixing it a little. So if you look at what Andrew presented, it was basically targeted at really big customers, and what Tim presented goes more to like the consumer. But if you are looking at big data, which the consumer sooner or later will probably be looking at, then in one way or another, because all data sets become big, and big is a question of definition, then we find that the difference between helping and leading is a very, very fine line. It's not always easy. And I think one of the issues that the consumer will have, and quite frankly, I think a lot of scientists deal with that too, is that you get a skewed view if you focus your search on the keywords that you know you're looking for. Basically, you're reinforcing your own preconceptions. Is there a way, and do your systems already include any way, of also looking for the things that are not in the data set? As an example, there has been a lot of scandal over fear of immunization due to just some incidents of side effects. And basically these episodic tales have spread over the internet and have spread in publications and did quite a number on what people conceive as the value of immunization versus the risk of it. And that, I think, has a lot to do with the fact that the data set was skewed, that the few data that were out there on side effects were elevated, and the lots of incidents where nobody had any side effect basically didn't get listed. So my question comes down to, do you have a way of finding the things that are not in your search but that are relevant to judging what the results of your search mean? So yeah, I shut up here.
Thank you. I think that both Andrew and Tim, you may have comments on that question.
Yes, certainly. I'll kick off. I think there are two questions in there. One is kind of like a mechanical one. How do you make a better search? And the other one is more philosophical. It's almost like I know the principles of reporting to people. And I think both are very interesting. In order to produce a better search, you have to have information that you can trust, and you have to have a means of weighting the information that you trust so that it is weighted so that the facts that are more reliable bubble up more than the facts that are less reliable. And it's something we see, and it's very interesting to us, is this kind of like whole strength evidence thing. What is the strength of evidence that X is true? And if you're trying to make a decision based on strength of evidence, you need to be able to collect that information and present it in such a way that people understand what is a reasonably good fact as opposed to speculation. And there's stuff we can do around search and algorithms, and probably Andrew could speak better than I can about that. I think there is a kind of like a responsibility in reporting angle as well. And I think that is something that we face across the board. If you're in the business of providing information to consumers, as broad as our broader organization we have a responsibility to check our sources, to validate our sources, and to make sure that the information that we report is honest and fair and unbiased. And I think that is something that we can bring to the consumer side, that sort of journalistic view of the world. I think will help to avoid, as Angus says, those sort of panics where somebody on the internet, I heard this morning, somebody on the internet had tweeted that the Queen had died, and suddenly that went everywhere. We want to avoid that kind of thing. You've got to have the ability to back up your sources and provide that strength of evidence.
Thank you. Andrew, would you like to add anything to that?
Yeah, I mean, I fully agree with Tim. I think that there are lots and lots of ways that you can discover unexpected connections. It's a big topic. Trust of information is really, really important, and that's a great example with the Queen this morning. I'd worried when I'd seen that. But weighting of information is also really, really important. So you might have a data source, particularly if you've got an integrated data source where you have some content that's of a very, very high quality and other content that's of a slightly lower quality, and it's really important to be able to differentiate those two things. The other one, though, is that is finding connections between information. So if we're talking about integrated data, you might have information on targets, you might have information on adverse events, you might have information on pathways. And traditionally, if all this information has been dotted around in lots and lots of different databases, there might be really important connections there, but you just miss them because the data isn't actually connected together. So that's one of the things that we're really behind some of the technologies that were implemented using CDF. There's some clever ways of exploring. We basically look at the data as a big graph of information that enables you to make new unexpected connections.
Well, thank you. I'm going to turn to some of the questions that have been entered by our audience. Some great questions here. For Dr. Logging in slide 8, could you please explain how new genomic technologies have enabled the NDA for drug candidate PGV001, and what was the benefit versus traditional approach?
Okay, I'm just pulling up slide 8. Yeah, so I'm not at liberty to sort of go in depth into the actual technique that we'll be utilizing within that clinical trial, but we will be sequencing the normal and tumors of individuals who have specific types of solid cancers, and applying omics analyses, obviously at the genetic level, but then bringing in a vast array of other data sets to best identify what specific vaccines should be generated for that patient. That's pretty much, I can't say too much more about that, unfortunately, but we're pretty hopeful that if this trial does work out, we'll be able to see a statistical difference between the treatment groups, which would get us pretty excited about moving forward with the next stages on it. But it's very big data driven, and again, the foundation of it is the genomics aspect.
Great, and here's a related question. How does the FDA view data like pathway inactivation in a filing? Does the submitting company have to show how pathway inactivation leads to a clinical outcome?
So some of the past experiences that I've had when we've taken this data to the FDA, it's actually been quite interesting. In my personal opinion, they've been very open to novel omics approaches. In the past, I've had follow-up questions come in where they've said, wow, we've not heard about this approach before. Can you please write a subsequent white paper on it and distribute it to us? Of course, then, you're sort of running frantic and, doing work till 2 or 3 a.m. in the morning because the FDA asked you to resubmit something. But it's very exciting. It's not like, we go to them and then we say, this is what type of analyses we're doing, and they say, well, we don't buy it. They're very open to it. Also, and to sort of specifically address that question, what we'd like and have done in the past is to say, at the molecular level, these pathways are changing, and that we feel that any sort of physiological effect that we're seeing in the clinic is related to those molecular changes. Now, we're all sort of built together based on certain aspects of biochemistry, and if one can show that there is a biochemical change and obviously propagated through the genetic level, then that is almost evident enough. Obviously there's still some sort of ultimate connection, whether you would bring in biomarkers or some other type of tested event that would say this is the reason why we're seeing this molecular change, and then we're seeing it at the physiological level. But those connections are being made, and again, the regulatory agencies seem very open to having that data submitted to them. Thank you.
Terrific. Thank you. Dr. Earhart, I'd like to invite you back into the conversation. Would you like to go further with your previous question or have a new question or comment?
Yeah, well, I'm tempted to do both. Actually, let me ask one question that goes to William. And so for him, he didn't just make fun of the increase in capacity and decrease in price and things like gaining genomic data. Actually, if you think about it, just the development itself, that it can be done that much more, pretty much brings it sooner or later to, let's say, the person on the street. So you also expect that the person on the street later wants to do that data analysis, which will be fun to do. We will probably see a lot of rather amusing things coming out of that, but maybe also a few interesting things. I think we have seen a few citizen scientists really making some progress for science where other people were stuck. Do you anticipate that the data analysis capacities will develop in the same direction as the data creation capacities? Right now in science, I feel that we are way behind in what we can do with the data versus with what data we can actually create.
Yeah, I definitely agree with that. Very much like, several hundred years ago, physics was an observational science, and now it's moved towards very much like a theoretical science. We are in the observational phase within biology, and we hope that we will gather all of this information. It will take a significant amount of time for us to be able to step through all of that information. I mean, if you've followed the sequencing of the first human genome, obviously when it came out in 2000, everyone was like, oh, well, we're done. It's going to be a new sort of age in genomics, and it's taken us 15 years to get to the point where we are at now. And I do agree with you. The interesting thing is I'm sort of I wax philosophical here, but we foresee a point where the sequencing will probably be close to free, and then a person will be sequenced many times throughout their lifetime. But what individuals will pay for is the interpretation of that information, and because it will be vast. And so that it's a very exciting point to be at, but we definitely need more eyes on target, as I stated in the one slide.
Yeah, I fully agree, and I think, I mean, I sometimes actually wonder what would have happened in physics when we still had a very deterministic world picture of the world, where you thought if you could only measure every particle in the world that exists, we could predict where the world goes 20 years from now. And lo and behold, quantum physics came in, and nothing of this predictability holds true anymore. Do you think that we will have a similar real big change in the way we think our data will lead us right now in as you said, in our observational stage, or do you think we will this time really succeed in predicting anything?
I think there'll be some low-hanging fruit that we'll be able to make predictions on, but again, it will take a vast amount of knowledge built upon how we're going to be analyzing this information. And again, what do we do with it? Experimental design is a very big key. Just being able to store all of the information and sort of distribute it, but then you have to come back to what is the distinct experimental design that you would want to do with that data? What are the questions that you're going to be asking of it? What ultimately do you want to do with the learnings from that information? Those are obviously key events that really need to be well thought out. Just about anyone can push terabytes of data around, but being able to apply them to applicable problems will be the key point. And again, I do agree with you. We don't have all the answers now. In some ways, we're still looking for the quote killer app, whether it is in data visualization or whether it's in some aspect of artificial intelligence that will allow us not to have to look through all of this data with our own minds. Like I said, eyes on target. Maybe we put some electronic eyes on target. Those types of things, we continue to think about, but it's definitely needed going forward.
Well, I really want to thank everyone for joining us, our terrific speakers and our large audience today. And there are a number of questions which I'll share with the speakers, and we'll try to follow up with you, questions that we didn't get to. And I'd just like to give each of you in turn a moment to say any final words you like. I'm going to start with Dr. Logging.
Again, thank you very much for the opportunity to talk about some of the exciting things that the field is doing. And again, I'd like to just sort of encourage everyone to think in that realm of we are approaching what we feel is sort of the beginnings of this golden age of being able to pull all this data together, but there's still a significant amount of work that has to be done.
Full Employment Act, right?
Exactly, thank you.
Tim, would you like to say a few words?
Sure. Yes, I agree with Will. I think we're on the brink of something really interesting, and I think we're figuring it out. I think what's really interesting and enjoyable is where we're figuring it out together, and it's a privilege to work with people like Will to kind of bring together the content, the technology, and the scientific know-how to kind of move this forward.
Thank you. Dr. Garrow? Taking a moment to unmute.
Sorry. I agree with the other two speakers that it's an amazing age that we're going into with the volumes of new data that we're now able to measure, particularly within the healthcare domain. But the other aspect of it is the way that technology, information technology, is evolving, because I think the two things go hand in hand, and it's going to be very, very exciting times to see how these two play hand in hand together.
Thank you. And finally, Dr. Erhard, I'm going to give you the last word, and then I'll just come back to say goodbye.
Well, thank you very much for everybody letting me be devil's advocate. I actually fully agree with all the speakers, and it was really good to just have at least a little discussion on where this is going and what we're going to see next.
Beautiful. Thank you, everyone. Have a lovely morning, day, or evening, wherever you are, and we'll look forward to seeing you in a couple of weeks for doing another webinar on the therapeutic relevance of venom. So you'll be seeing an email about that shortly. Thank you so much. Take care.